Hello and welcome to Teaching Bio. In this video, we're going to be looking at exchange with the environment, and ultimately we're going to be talking about surface area to volume ratio, adaptations to maximize exchange, and we're also going to be looking at insect, plant, fish, and human exchange systems. So this is designed to cover most exam boards for AS and A-level biology in mind. Okay. So we'll start off by having a quick discussion about exactly what types of things need to be exchanged and what we mean by exchange with the environment. So the first thing you'll probably be aware of, what you might be thinking of, is respiratory gases. So that's things like oxygen and carbon dioxide. So obviously we inspire oxygen, so we breathe in oxygen and breathe out carbon dioxide, which needs to be exchanged. As well as that, things like glucose, fatty acids, minerals, ions, and so on and forth also need to be exchanged. And this type of exchange occurs more in a homeostatic fashion. So the control of blood glucose levels, the control of water potential, and sort of reabsorption of things like proteins and ions at the kidneys. And again, thinking about sort of excretory products and things that we need to sort of get rid of. So urea is a major waste product that we get rid of in the urine. And again, carbon dioxide, as we talked about before, is one of those respiratory gases that's a waste product that we need to remove because it's acidotic and can cause devastating impacts to our blood pH if we aren't able to get rid of it efficiently. So how do these sort of exchange mechanisms occur? Well, they occur by transport mechanisms across whatever relevant membrane it might be. So for example, in the alveoli, in terms of sort of human gas exchange, or across the gill lamellae, if we're talking about fish. And again, these all have their different respective mechanisms involving a mix of diffusion, active transport, facilitated diffusion, osmosis, etc. Okay, so we're going to start off by talking about surface area to volume ratio. So surface area refers to the area outside of a cell, and the volume refers to the area within the cell. Therefore, the surface area to volume ratio is the ratio outside of the cell to the volume inside of the cell. Okay, so here we've got an example sort of table of how the surface area to volume ratio decreases as we sort of change the size of a cube here. So the cube is getting bigger, the surface area is getting bigger, the volume is getting bigger, and therefore the ratio between the two, so i.e. the ratio from the surface area to the volume will decrease, okay? Because there's less surface area respective to the volume left. So as the size increases, the surface area to volume ratio decreases, and that's the fundamental process that you need to understand, or the fundamental concept, sorry, that you need to understand. However, this does not mean that the surface area in and of itself is decreasing. It's the ratio that's changing. Big organisms will still have a big surface area. As we can see here, an 8 centimeters cube, an 8 centimeter cube has a larger surface area, 384, compared to a 2 centimeter cube. However, its surface area to volume ratio is much smaller compared to the 2 centimeter cube. Okay, so the larger the organism, the smaller the ratio, but the larger the surface area value in and of itself. Therefore, given us as humans and sort of larger mammals, simple diffusion is not enough to maintain these exchange systems. So a simple cell like an amoeba can rely on simple diffusion for sort of exchange to occur, but as the organism gets bigger, we need to develop specialized exchange mechanisms, and as a result we have specialized exchange organs and organ systems to allow that. And that's not just limited to us as humans, but also things like other animals, fish, insects, etc. And ultimately the point of having these exchange systems is to maximize the rate of diffusion. Okay, so to ensure a faster rate of diffusion, to allow for a faster rate of exchange, so that we can maximize how much oxygen, for example, we're taking up, or how much carbon dioxide and waste products we're getting rid of. Why is it important then to think about the surface area to volume ratio? Well, again, as we've sort of discussed, it's to sort of maximize the diffusion of things like nutrients and oxygen in, and also maximize the diffusion of waste out. Having too few nutrients would mean that cells cannot grow, and waste products building up can be toxic. And we talked about carbon dioxide before. And as I mentioned, carbon dioxide is a weak acid when it dissolves in water, and it lowers the pH of blood, which can cause acidosis, and that's very dangerous. Okay, so pause the video and have a go at this question here, which talks about seals, and it's having a look at how the body shape of this seal is adapted to living in a cold environment. Okay, hopefully you've had a chance to attempt this question. So the mark scheme for this was to effectively state that the seal will have a small surface area to volume ratio, which makes sense because it's a large organism. And as a result of the small surface area to volume ratio, less heat will be lost across that surface area, okay? Because the ratio relative to it, the ratio relative to the volume is smaller. So it minimizes the amount of heat loss, therefore meaning that more heat will be retained. And in the context of this question here, that's important for these types of seals so that they're adapted to live in these cold environments. By having a small surface area to volume ratio, less heat energy will be lost in respiration, so more heat energy will be retained, therefore meaning that it can survive in a cold environment. Okay, so here's a slightly more challenging question. Pause the video and have a go and see if you can answer this one. Okay, so this question talked about the amniotic sac. So the amniotic sac is the sac in which a fetus develops in the womb. This sac is important to allow for the development because it contains things like nutrients, ions, respiratory substrates, and hormones. So that's things like glucose, etc. So the question asks you to use your knowledge of exchange systems to suggest and explain how the sac is useful for the development of fetuses. So these are some things that you might have thought about. The fact that the sac contains these sort of ions, nutrients, and hormones means that it can directly diffuse into the fetus by simple diffusion. Note that simple diffusion is the mechanism that will be used at this point because it's a fetus, which is effectively just a ball of cells. We're not told how developed the fetus is, but we can just assume it's in the earlier days, given that we're calling it a fetus. So as a result, it can rely on simple diffusion mechanisms. And that's because it is small, therefore it will have a large surface area to volume ratio. So this is sort of the opposite of the previous concept we were talking about. No exchange systems have been developed yet, or those exchange systems are still in development. So things like the cardiovascular system, so the heart and the lungs are still being developed at this point. And things like the lungs aren't developed until about 32 weeks in a fetus anyway, so it won't be relying on that. So predominantly it will use the nutrients within the amniotic sac to allow for its development, because these nutrients, hormones, ions, respiratory substrate, glucose, etc., will ultimately be used for the growth and mitosis of the cells of the fetus. Okay, we're now going to move on to talk about insects and the exchange systems in there. So terrestrial insects effectively live on land, and it's important for these insects to maintain water because water can be lost very easily from their body. As a result, insects have developed many features to try and minimize the loss of water. So for example, insects have this hard waterproof exoskeleton which will physically prevent the loss of water. And also insects are, obviously they are quite small creatures if we think about them compared to us, but then comparing that to something like a simple cell, they are much bigger. Therefore, they will have a relatively smaller surface area to volume ratio. And again, similar concept as we were talking about with the seals before, but in this case, it's less so to do with the amount of heat loss, but more so to do with water, okay? So whenever you see insects in the exam or you're thinking about insects, always think about how water is maintained, and that same concept applies to plants as well. So insects have small surface areas compared to mammals, but as we said, they are large in respect to simple cells, and therefore they've got that small surface area to volume ratio. As a result, they cannot use simple diffusion to allow an exchange. And again, simple diffusion is not enough to allow for them to retain water, so they develop their mechanism. So ultimately, this is how insect exchange occurs. They've got these specialized systems, kind of similar to human exchange systems. So they've got these openings called spiracles, also a bit similar to plants if we think about the stomata, along its exoskeletal surface. That then splits into the trachea. The trachea splits into the tracheoles, which have many branches that then connect directly to the muscle fibers of the insect to directly supply oxygen so that respiration can happen and it can generate ATP for muscle contraction to allow it to move. This is another sort of image of the tracheal system, which again, as we talked about, that was the spiracles. There's sort of exoskeleton on the outside. Here's the trachea branching into the tracheoles, attaching specifically and directly to the muscle cells or muscle spindles. So the spiracles, they look a bit like this. So these small openings on the surface, and these are for the diffusion, i.e. the entry of oxygen and the exit of carbon dioxide. The trachea, as we talked about, so these are the trachea here. The trachea is effectively a long network of tubes, and they are made of these rings of chitin, or chitin, if you want to call it that. But these rings of, chitin, are deposited in the walls of the trachea here. And the point of these rings is to prevent the trachea from collapsing. Okay, so it gives it durability and strength, meaning that gases can freely travel in and out of it. Then the trachea connected to these tracheal tubes. So here we can see the structures of them. These are much smaller tubes and are very highly branched and also very thin. So again, as you can see from like this terminology here, these are the kind of words that you want to be using in the exam to be describing the tracheals compared to the trachea. Because of the fact that they are highly branched and because they are very thin, it means that diffusion can happen faster. The tracheals are also directly connected to those muscle spindles, cells, tissues, etc., so that exchange can rapidly happen because it's right sort of next to it. Okay. Another key thing to sort of think about is the movement of fluid that happens within insects. And this is an example question that was asked once. So here we've got a stem about insects. So in insects, air is supplied directly to the tissues through a system of tubes called trachea, okay, which branch to form smaller permeable tubes called tracheals. Okay, we've established that. And the diagram shows the change that occurs in these systems and the tracheals supplying muscle fibers during flight. So at rest, we can see that there's some liquid here within these tracheals, and then during flight, the liquid decreases, okay? So during flight, the liquid in the tracheals passes from the tracheals into the muscle fibers. So as it's flying and using energy, that fluid is going to move into these muscle fibers. So here were the questions that were asked. So how does this change benefit gas exchange in the insect? So by having the fluid move sort of towards the muscle, this will increase the surface area. Okay, so if we think about it, this is the amount of surface area for exchange. If that fluid decreases, there's more surface area for exchange now. So larger surface area meaning faster rate of diffusion, so there's more gas exchange. The next question talked about substances which are produced during metabolism cause the liquid in the tracheals to pass into the muscle fiber during flight. So this is saying exactly how that fluid might move. So these substances are probably going to reduce the water potential of the muscle tissues. Again, because it's a liquid, it's probably going to move by osmosis. So by lowering the water potential, we create a water potential gradient. There's a higher water potential in the tracheals compared to the muscle tissues. So the liquid in the tracheals diffuses down the water potential gradient into the muscle tissue by osmosis. Okay, and the last question here was to suggest why a muscle fiber in which the tracheals are restricted to the outside of the fiber cannot be larger than 20 micrometers in diameter. And again, it might seem like a bit of a weird question when they throw things like these numbers here that you aren't too comfortable with, but just go back to your basics, which is about size and about surface area to volume ratio. And if they ever give you something about diameter, or they give you things to do with the size, it's always going to be related to this. So they cannot be larger than 20 because if it's more than 20, it's probably too big. And so it limit the rate of exchange, i.e. the rate of diffusion, because it will increase the diffusion pathway. Okay, so they need to be smaller than that so that we can maintain the fastest rate of diffusion that we can. Okay, so we're going to go through a summary of insect exchange now, and this is sort of the standard exam answer you should be writing. So oxygen diffuses down its concentration gradient from the outside atmosphere into those spiracles, those openings we talked about, then into that trachea lined with those rings of chitin for support, and then into those tracheals, which are those smaller branch tubules that connect directly to the muscle fibers and spindles directly. So yeah, the tracheals are directly into the muscle fibers. Therefore, we allow for that rapid diffusion of oxygen. They go into the mitochondria of the muscle cells, which produces ATP by respiration, and that releases carbon dioxide. Once high levels of carbon dioxide are detected within the insect, this induces a mechanism called abdominal pumping. Abdominal pumping, which as the name suggests, is the contraction of the muscles of the abdomen of the insects, puts pressure on the tracheals. Okay, so it's squeezing the tracheals, it's increasing the pressure of the tracheals because it's contracting once those carbon dioxide levels are detected. As a result of that contraction, this will increase the pressure of carbon dioxide within those tubule systems. Okay, so we've squeezed the tracheals, so we've increased the pressure. Because of that pressure being increased, we've created a pressure gradient. And as a result, this forces out carbon dioxide out of the sort of out of the exchange system through the spiracles back into the atmosphere. So oxygen enters by concentration gradient and carbon dioxide leaves by a pressure gradient. Okay, so carbon dioxide out, down its pressure gradient. So very important to remember that oxygen entry is concentration, but carbon dioxide exit is pressure in insects. Okay, so again, we've kind of briefly mentioned these, but they like to ask questions about how sort of systems might be adapted. So again, we've talked about the spiracles. Again, the fact that these can open and close is probably linked to being able to conserve water. Abdominal pumping, we've talked about that and how that will maintain that pressure gradient for the carbon dioxide to move out. And also it will maintain a concentration gradient for oxygen to come back in because it allows for the exchange of those two systems. We've talked about the rings of chitin to prevent the collapse of the long tube, the trachea. We've talked about how the tracheoles have thin walls and are highly branched so that, again, they have a large surface area, short diffusion distance. And again, we've talked about the fluid that can move. And by having the fluid move, we can increase that surface area so that we can have a faster rate of diffusion. And whenever you talk about insect adaptations, always link your answers to water loss and you'll be guaranteed the marks. Okay, so pause the video and have a go at this exam question and we'll have a discussion of the answer in a second. Okay, so this question sort of talks a bit more about abdominal pumping. So the question states that abdominal pumping takes place during vigorous activity in insects. This causes regular squeezing of the tubes of the gas exchange system. So these tubes are referred to as the tracheoles. A scientist investigated the effect of abdominal pumping on the pressure of the tubes and the volume of carbon dioxide released by the insect. And as you can see, as the carbon dioxide release sort of increases, that pressure will it sort of decreases, then increases, decreases, then increases, and that's related to the abdominal pumping. Okay, so you have to describe and explain these results. So these are the marks that they wanted in the mark scheme. Number one, abdominal pumping / pressure in tubes is linked to carbon dioxide release. Again, that's nice and obvious point. The fact that abdominal pumping must be linked to CO2. This pumping raises the pressure in the body of the insect. And as a result, it will move out the carbon dioxide. They allowed air, but I don't think air is probably the best answer. I think talking about carbon dioxide is probably more specific. So carbon dioxide moves down the pressure gradient is probably the better answer here. Okay. And interestingly here, it says to reject concentration gradient, okay, because that is incorrect in the context of carbon dioxide exchange in insects. It's to do with pressure gradient, not concentration gradient for carbon dioxide. Okay. So we're going to switch gears and we'll now talk about plants. So plants use the stomata for gas exchange, which are also openings. Oxygen diffuses in and down its concentration gradient and carbon dioxide diffuses out and down its concentration gradient. Okay, so in plants, oxygen and carbon dioxide both rely on a concentration gradient, not a pressure gradient. Water vapor will also transpire from the stomata. And this opening and closing of the stomata will be controlled by guard cells, and they're sort of, so we'll talk a bit more about guard cells in a second, but they will change depending on the amount of water there is and become flaccid or turgid. So this is a cross section of a leaf. And as you can see, you've got the stomata, which is the opening. Okay, so these aren't the stomatas. The stomata is the literal opening between those guard cells. Okay, and it's the swelling or shriveling of the guard cells that controls the degree of opening of the stomata, if you like. And then again, there's these air spaces which are important to allow for that exchange system. We've got the waxy cuticle, which is an important adaptation to allow for this waterproofing mechanism as well. And we've got different types of mesophyll. You've got palisade mesophyll and spongy mesophyll, and the two layers of the epidermal tissues. Okay, so the stomata, we talked a bit about guard cells. So effectively, stomata need to be open. We need to have that space to allow for the entry and exit of those gases. And they will be open during the day to maximize the rate of photosynthesis within plants. So this will be the entry of carbon dioxide for photosynthesis. And then the stomata often close at night to conserve the water, and this is when plants are typically respiring. So this is when they're producing carbon dioxide instead of taking in the carbon dioxide so that the carbon dioxide can be removed. Okay, so in terms of plant adaptations then, again, things to talk about, things like the fact that there is a large internal surface area, again, faster rate of photosynthesis, the fact that we have many stomata helps to maintain that gradient, and the fact that the leaves are thin means that there's a short diffusion distance. And with photosynthesis, with plants, it's always good to talk about photosynthesis because that's ultimately what these adaptations are for. Okay, so pause the video and have a go at this challenge question here, and we'll have a discussion of the answer. Okay, so this question states that ABA or ABA is a substance that prevents the stomata from opening. Suggests 4 reasons why this might be advantageous to plants. Okay, it's a slightly tricky question, but effectively this is asking you to state why the stomata being closed might be good. And again, like I said before, you've got to think about these answers often involving plants in the context of photosynthesis and gas exchange and also water. So ultimately, it's about the conservation of water. So by conserving water, and note that it was to suggest 4 reasons why, so you've got to expand on your points. Conserving water allows for more or faster or greater photosynthesis. Therefore, the plant can produce more ATP/glucose. If closed, there'll be very little water being lost by transpiration. Therefore, more water can be conserved for, again, photosynthesis. And then the photolysis of water, again, is the integral steps of photolysis, the first step of photosynthesis. Again, conservation of water lowers the overall effect of the rate of transpiration, so there'll be less transpiration occurring. If closed, there is a chance that more respiration can happen because it can retain those gases, therefore produce more ATP. Conservation of water keeps the plant upright or turgid, which is again a good point if you think about how plants, obviously you can't have the plant being floppy and falling over because then it won't be, it won't function properly, so it's got to stay upright. And again, slightly more rogue answers, but thinking sort of about physically, other sort of adaptations, or the sort of advantages this might have. The fact that the stomata are closed might mean that bacteria or things like pathogens or spores in the air currents are less likely to diffuse in and cause harm to the plant. Okay, we're now going to move on to talk about fish. So fish have a waterproof outer coating and also have a small surface area to volume ratio. In order to maximize the rate of diffusion, fish have developed gills, and gills have a fantastic mechanism of action to sort of maximize the rate of oxygen uptake for fish as they live in water. So the gills are located behind the head of the fish, and they're protected by this thing called the operculum. So this is a sort of image of the gills. The gills themselves are made up of many gill filaments, and those gill filaments, so here the gill filaments, they are then made up of gill lamellae within themselves, and all of those gill lamellae and gill filaments are connected by gill arches, okay? And effectively they function via something known as a counter-current exchange system, which we'll talk about in a minute. So in terms of how fish actually ventilate, if we like, so we'll take it step by step. So ultimately, first, water's got to obviously enter the mouth cavity of the fish. The fish is in water, therefore water's probably going to enter. Once the water enters, the fish will close its mouth. If it closes its mouth, the floor of its mouth, which is called the buccal cavity, is going to rise. The rising of that floor of the mouth reduces the volume in the mouth, therefore increases the pressure. As a result, that increased pressure causes the operculum to open, and that forces the water back out across the length of the gill lamellae down the pressure gradient. And it's at this point that we have our countercurrent exchange mechanism happening. So as that water flows across the lamellae, and it can only flow because of that increase in pressure, the oxygen from the water can diffuse into the blood via the countercurrent flow system. So how does this work then? So water and blood are going to move in opposing directions across the length of the gill lamellae, and the concentration of oxygen is always higher in the water compared to the blood. So as a result of that, there will always be diffusion of oxygen from the water into the blood down the concentration gradient. So ultimately, the purpose of the countercurrent flow is to maintain an oxygen concentration gradient. So again, this can be a common question in exams. So if we think about this being a 6-mark answer, sometimes this could be a 2, 3, 4-mark answer, who knows? But this is the sort of the best answer you could write. So explain the countercurrent flow system, water and blood flow in opposite directions, that'll get you a mark. The concentration of oxygen is always higher, next mark, in the water than the blood, and so oxygen always diffuses down, next mark, its concentration gradient into the blood, next mark, across the length of the gill lamellae. They love this marking point here about being across the length of the gill lamellae, so always throw that in, and this helps to maintain an oxygen concentration gradient. Okay, so if we think about oxygen and temperature then, and more specifically sort of thinking about water temperature, how that might affect ventilation in fish, as the water temperature increases, we can see that the ventilation rate increases. And on this graph here, we can see that as the water temperature increases, the solubility, so how much oxygen is dissolved within a kilogram of water, actually decreases. So this suggests that oxygen solubility, which is ultimately can be taken to be the concentration of oxygen within the water, will decrease as the temperature increases. As a result, the fish has mechanisms whereby it will increase its rate of ventilation to accommodate for that change. So to accommodate for this, the rate of ventilation increases so that more water can pass over the gill lamellae, okay? More ventilation will mean, again, thinking back to the mechanism of ventilation, will mean that more water will enter. So therefore, there'll be more of an increase in pressure once it closes its mouth and raises its buccal cavity, so that more water can be forced across the gill lamellae, so that oxygen can diffuse into the blood via the countercurrent mechanism and therefore be reabsorbed. In terms of fish adaptations, you should always think about talking about oxygen. So again, we've talked about the gill lamellae, but the fact that we have the fact that fish have so many of them means that there's a faster rate of diffusion, so the oxygen can sort of constantly be absorbed. These lamellae filaments are thin, which provides a short diffusion distance, and the fact that there's that countercurrent flow, which again functions to maintain that oxygen concentration gradient along the length of the gill lamellae. Okay, so very important to remember this phrasing here, along the length of the gill lamellae. Okay, so pause the video and have a go at this maths question here, and we'll have a discussion of the answer in a second. Okay, so for this question here, you had to calculate the volume of water that would have to pass over the gills each hour to supply the oxygen required by the fish. So it can seem a bit confusing, this answer, but it's not as bad as it seems. Ultimately, all you have to do is manipulate these numbers in the right way. So you've got to do 0.4, which is the mass of the fish, times that by the oxygen that will be required by the fish per hour. Okay, so that's in kilograms, so that'll give you the oxygen needed per kilogram. Okay, then you've got to work out how much volume will actually be absorbed per decimeters cubed. Okay, now interestingly, these units they've given in centimeters cubed, which can make everything a bit confusing, but ultimately take the units that they've given you as your main marking, as your main reasoning for your answers. So all you've got to do is 0.4 times 90 and divide that by 7. That's it. 0.4 times 90 divided by 7. And that'll give you that answer there. Okay. So here's a challenge question for you. Pause the video and have a go, and we'll have a discussion of the answer in a second. Okay, hopefully you've had a chance to have a look at this question. So here, this question is talking about mackerel. So mackerel live in the surface waters of the sea, and toadfish live on the seabed in deep water. Mackerel intake greater concentrations of oxygen compared to toadfish, and the question is asking you to suggest why. Okay, so why is the mackerel taking in more oxygen compared to the toadfish? And the only difference between them two is that mackerel are on the surface and toadfish are on the seabed. Therefore, the logical assumption or inference you've got to make is that the mackerel, as a result of being on the surface, is taking up more oxygen because there's probably more oxygen on the surface of the water compared to the seabed. Okay, so the reason why there's more is because it's at the top, and if it's at the top, it's close to the air currents, and if it's close to the air currents, it means that it's closer to, you know, the higher concentration of air and therefore oxygen. Okay, so mackerel is at the top, so it's closer to the air currents, so a greater concentration of oxygen can diffuse into the mackerel. Toadfish is at the bottom, so its source of oxygen is mainly limited by the sort of the water there, and also by plants. And there's no sort of air currents there, so there's none of this constant supply of oxygen that might be given for the mackerel compared to the toadfish. Okay, so no such hardly any currents at the bottom. So less oxygen for toadfish, more for mackerel. Okay, we're now going to talk about human exchange systems. So here we've got the lungs, and I'm sure you're all familiar with what the lungs do and what they're for. But again, they're made of these spongy materials so that they can inflate and expand to allow for the expansion of air, and then also deflate to allow for the expiration. Okay, so we'll walk through the structures of the human ventilation system then. So we've got the trachea, which is this sort of tube here. And again, similar to, so we talked about the rings of chitin in insects, but in humans, there are sort of cartilaginous rings, which again have the same function, which is to prevent the collapse. Okay, so there's this long tube, which is the trachea, and that branches into the right main bronchus and the left main bronchus. Interestingly, the right main bronchus is slightly higher up than the left main bronchus. The bronchus then branches into the sort of these many bronchioles. Okay, so these are smaller and branched versions of the trachea, and these are lined with ciliated epithelium, okay, which are covered with mucus, which are very important to allow for the maximization of the diffusion gradients and to allow for that transfer of gases. Again, the bronchioles, which are these terminal branches here, they're even smaller and they're also ciliated, but they generally don't really have mucus glands. The mucus glands are limited more to the bronchi structures here, the right and left main bronchus. Okay, and then so here we've got lobular bronchiole, terminal bronchiole, respiratory bronchiole. You don't really have to know this level of detail, just being aware that they are called bronchioles is probably enough. And then these connect directly to the alveoli, which are composed of ducts and sacs. So the alveoli, which look a bit like these grape-like things here, they are very small cavities that are surrounded by thin elastic tissue. That elastic tissue is very important because it allows for that stretchiness to accommodate for the filling of air. And it also contains very thin squamous epithelium cells, which are directly tied to a capillary network. So those very thin squamous epithelial cells on the surface of the alveoli means that there's very rapid and very quick gas exchange between the alveoli and the blood vessels, because there's a very short diffusion distance that needs to be traveled because they're virtually sort of connected, okay? So if we talk through exchange in the alveoli then, and again, these are our squamous epithelia that we were talking about before. So ultimately, we breathe in oxygen, oxygen is inhaled, it'll move down the trachea, bronchus, bronchioles, and it'll fill the alveoli, okay? So all the oxygen is entering into the alveoli stacked here as it expands. Eventually then, the oxygen will enter, it'll diffuse into the red blood cells here, and they'll be moved along and they'll be oxygenated and form oxyhemoglobin, etc., and then go off. And the other blood cells that have had their oxygen already offloaded at the peripheral tissues will come back to the lungs to deposit out the carbon dioxide that they've picked up from peripheral tissues to exchange. Okay, so oxygen is sent off in these capillaries here, and the deoxygenated blood will come and deposit all the carbon dioxide, which will diffuse down its concentration gradient because there's a higher concentration of carbon dioxide here compared to here. So carbon dioxide diffuses down its concentration gradient from the red blood cells to the alveolus, therefore being unloaded, and eventually there'll be a buildup of the carbon dioxide concentration here so that they can be forced out. So it'll be high here, lower here, fills it with here, so that eventually it increases here, and then it can be expired out. Okay? And again, as we talked about, there's this simultaneous diffusion of oxygen down its concentration gradient, meaning it can be loaded or bound to hemoglobin and moved to peripheral respiratory tissues. Okay. Fine. So now we're going to talk about ventilation. So again, there's these sort of rules and facts that you should remember about ventilation, volume, and pressure. So we have our external and internal intercostal muscles. So the contraction of our external intercostal muscles is very important for inspiration. So by contracting the external intercostal muscles, we can pull our ribs up and out, which is needed for inspiration, which makes sense if you think about taking a deep breath in. Your ribs have to obviously move up and out to allow for that expansion of volume so that we can have that entry. In expiration, there is contraction of the internal intercostal muscles, which, as you might have worked out, to the opposite of what is happening in inspiration, where instead the ribs are going to move down and in, okay, so that we can expire those gases out. In inspiration, the diaphragm muscle is going to contract, which functions to increase the thoracic volume and decrease the pressure, so that we can create a pressure gradient to allow for the entry of oxygen. And in expiration, the diaphragm muscle relaxes and the diaphragm flattens so that we can force those gases out. So this summary table here might make things a bit easier to visualize in terms of what's happening in inspiration and expiration. It's important to note that this is known as an antagonistic interaction because they do the opposite effectively in each of these stages. So the external intercostal muscles contract in inspiration, and the internal intercostal muscles relax in inspiration, and the opposite happens in expiration. So in inspiration, we're going to have the movement of the ribs up and out. We're going to have contraction of the diaphragm, which will flatten it, therefore increase the thoracic volume and decrease the pressure. And in expiration, we'll have the ribs moving down and in. We'll have relaxation of the diaphragm, which will therefore cause it to become more domed so that we can decrease the volume of the thorax and increase the pressure to allow for the exit of gases, okay? So we've got to have this decreased pressure in the thorax so that gases can come in, so they can diffuse down their pressure gradient. Whereas here, so by gases in the inspiration, obviously I'm talking about oxygen, and whereas here we'll increase the pressure, so we'll have an increase in carbon dioxide pressure so that we can, an increase in thoracic pressure so that we can create a pressure gradient for carbon dioxide to be forced out. Okay, so human adaptations, again, they love to ask questions on this. So things like the fact that there is this constant simultaneous diffusion of oxygen in and carbon dioxide out. Ultimately, this maintains that concentration gradient, which is very important to do. The fact that we have many alveoli means that there's a large surface area, so it's faster. The fact that there's that thin one-cell thick membrane of those squamous epithelia, it's the endothelium, not epithelium, endothelium, means that there's a short diffusion distance, so there's a faster rate of diffusion of gases. And the fact that each alveoli is surrounded by this, rich network and supply of capillaries means that, there's always this constant supply of blood so that we can maintain these exchange systems. The fact that there's a large difference in the concentration of carbon dioxide and oxygen means that, again, we can maintain those gradients so that carbon dioxide is always diffusing out and oxygen is always moving in. Constant ventilation mechanisms, again, help with maintaining those gradients. Okay. So sometimes I like to ask some maths questions. So this question is about pulmonary ventilation. So pause the video and have a quick go at this question using the formula there, and we'll talk through the answer. Okay, so pulmonary ventilation, which is measured in centimeters cubed per minute, is equal to the tidal volume times by the breathing rate. And the question asks you to work out the breathing rate in minutes for a person who has a tidal volume of 1,000 centimeters cubed and a pulmonary ventilation of 300 centimeters cubed per second. So for this question, we've got to rearrange the equation to work out the breathing rate. So breathing rate will therefore be equal to the pulmonary ventilation divided by the tidal volume. Okay, then all we do is we plug in these numbers. However, note that the pulmonary ventilation in this question is given per second, whereas the equation requires us to use per minute. So all we've got to do is do 300 times 60, because there are 60 seconds in a minute, and then divide that by 1,000. And that should give you an answer of 18 breaths per minute, which is normal. Okay. Sometimes in exams, they like to talk about lung diseases as application questions, and generally these are the types of answers or the sort of vibe of answers you should be writing. Things like there being a smaller surface area of the alveoli, the fact that the alveoli might be less elastic, or the fact that the alveoli might be thicker. And again, a lot of the damage in lung disease is to do with the alveoli. But it can also be higher up in the trachea if we think about things like asthma and so on. That's all to do with narrowing of, not the trachea, the bronchus. But ultimately, these changes will lower the rate of this exchange and therefore lead to impairments in gas exchange. So here is a model answer from the sort of new papers on asthma. So what they want you to say is things that the muscle walls, okay, so very important to know the muscles of the bronchial, so not the trachea, the bronchioles contract. The walls secrete more mucus. And again, remember we talked before about how some of the cells that are lined with that mucus, as a result of that constriction, so that's bronchoconstriction, that leads to reduction in the diameter of the airways, therefore reducing the flow of air and henceforth reducing the rate of diffusion. Okay, so pause the video and have a go at this challenge question here, and we'll have a discussion of the answer. Okay, so this question is talking about emphysema, which you might not be familiar with, but that's okay. So emphysema is a lung disease that damages the walls of the alveoli. Doctors observed this led to a decrease in the pH of the blood of a patient with emphysema. Use your knowledge of ventilation to explain why. So if you ever see a question involving pH in the context of lung disease, it has to do with carbon dioxide. So this suggests the reason why the pH is decreasing, i.e. the pH is becoming more acidic, suggests that there's an increase in carbon dioxide, meaning that there's an impairment in the gas exchange. And that's effectively what the answer is. So the question says that it's damaged the walls. So altered walls or really damaged walls means that the alveoli will probably have a smaller surface area. Therefore, that will lower the rate of diffusion of carbon dioxide out from those red blood cells into the alveolar sac and out of the lungs, meaning that it takes more time for that carbon dioxide to leave, okay? So we've limited the diffusion of carbon dioxide, so it takes longer for carbon dioxide to leave, meaning that the carbon dioxide will be in the blood system for longer, so there'll be more of this buildup of carbon dioxide, which ultimately will lower the pH because carbon dioxide is a weak acid. Okay, thank you very much for watching. I hope this video was helpful, and let me know if you have any questions.